Right, so last class, we ended with an example of how you use PCR and agarose gel electrophoresis to find whether or not you've successfully created an XX organism, so 2 X chromosomes, no Y chromosome, that contains just one extra gene. So normally an XX individual will be female, but say you insert the SRY gene from a male into an XX organism. And then you saw the picture, mouse dangling from a bar. You could see its ventral surface. You could see its genitalia. If you're a mouse expert, you could see it's a male, at least according to its external genitalia. The question here is this mouse functionally male? Does it produce fertile? Is it fertile? Does it produce sperm? And are the sperm able to fertilize eggs? Unfortunately, no. So it looks like a male, but with just the SRY gene. So that's the testis determining factor, which we saw at the start of last class. This is a gene that's required. You have to have it to be a male. Unfortunately, you have to have a few other genes to be a fertile male. So there are other genes on the Y chromosome. So SRY comes from the Y chromosome. It's a gene on the Y chromosome. But there are a lot of other genes that are involved in spermatogenesis that are also on the Y chromosome. So really, if you want to be a male and be able to produce offspring, you have to have more than SRY. You have to have SRY plus at least one other gene that helps you produce functional sperm. By the way, while we're going through class, if any of you are still on the old networks, our Guide on the Go is wandering around helping people get connected to Idiograms, the new wireless network, in advance of our exam on Friday. So if anybody needs that support, that's going on at the same time we're reviewing. Other questions? Yeah?
Pertaining to this, if you deleted the SRY gene from the Y chromosome, then what would happen?
So this all started with my comment. One of our students, posted that a deletion causes feminization, I think was the situation, right? So if you have an X chromosome, if you have a Y chromosome, so you've got an X and Y, the Y chromosome is much smaller than the X because it's literally falling apart. Over millions of years that our Y chromosome has existed, it constantly suffers genetic deletions. So if guys, eventually our Y chromosome, it's gone. It's jettisoning DNA every generation. Little small deletions on it means our Y chromosome is getting smaller and smaller. So one thing to wonder is what happens when our Y chromosome totally disappears, but that's maybe for another class. So normally the Y chromosome has SRY that makes a male. What happens if you have an XY individual that's missing SRY? So say there was a deletion on the Y chromosome of the part of the Y chromosome that contains SRY. What's the sex of that individual? They've got an X chromosome, they've got a Y chromosome. So what sex? Male or female? So if the Y chromosome, as we saw last class, is sufficient to make a male, you move it to an X chromosome, makes a male, although infertile mouse. When you delete SRY, you get a female. So what we talked about last class was about sufficiency. Is SRY sufficient? You put that gene into an organism that's never had it before, what happens? Changes that individual anatomically into a male. Not a functional male, but anatomically. This experiment tests necessity. So this means SRY is necessary to be a male. If you don't have it, through mutation, deletion. You can have your Y chromosome still, but if it's missing that one gene, or if that one gene, SRY, is mutated so it doesn't function properly, then you're not a male. And there are naturally occurring mouse mutants that exhibit this. So this is how this experiment was done. It wasn't an experiment that was done. Scientists were looking at lots and lots of mice, and they were genotyping them, and one day they found a mouse that had a mutation in its SRY gene. So it had a Y chromosome, but it was an anatomically female mouse. And in that case, those mice are fertile, females. This sort of got scientists, this is a hotly debated topic that I'm about to mention still. This kind of got scientists starting to think about the idea of, is there a default sex? So if there's a default sex in mammals, which is it? Males or females? That's what a lot of scientists thought, was female is this default sex, because it takes one extra genetic element to become a male. You have to have SRY. If you don't have SRY, then you're female. So this is hotly debated. It's more of a concept than a fact. It is a fact that if you don't have SRY, you're female, but the idea of there being a default sex is fraught with all sorts of peril, so that's why it's debated. More of the sociological or philosophical impact than a scientific impact. I think that pretty well covers, so necessity and sufficiency. To test if something is necessary, a gene is necessary, you get rid of it, you delete it, you find a mutation in it, you find a naturally occurring deletion that gets rid of that gene, and then you look what the effect is. To test sufficiency, you take that gene and move it into an organism that has never had it before, and you find out what the effect of adding a gene in the naive context does. Necessity and sufficiency is one topic we talked about in class this time. So related questions, follow-up questions, new topics? Yes, ma'am.
I was confused in a PCR reaction. How do we make sure that the probe doesn't hybridize to itself instead of hybridizing to the chromosome?
Oh, so FISH, so let me interpret your question. I think what you're asking, if I understood correctly, is in a FISH experiment, we have, as we discussed, maybe one double-stranded chromosome. And the first step in FISH and in PCR, heat it up, melt the DNA, denature it, so that it's single-stranded. That means something else can come in and base pair with the DNA sequence that we're trying to identify. So this is the same in PCR and FISH. They share a common, this is common to both of those techniques. You start with double-stranded DNA, you heat it up, then you cool it back down. And we talked about how you force, one of you asked a question, how do we know that this chromosome, when you cool this mixture back down, the chromosome doesn't just base pair with itself again? We put a lot of either, in PCR, primers, or in the case of FISH, probes in. So you have loads and loads of these tiny double-stranded primers or probes. In this case, I'm drawing probes, double-stranded, hybridized to each other. Short little DNA sequences relative to the size of the chromosome. So this is to make sure that when we start cooling the mixture back down, one of those single-stranded probes, after they're heated up, will force itself into the chromosome and base pair with the chromosome, where it has a complementary DNA sequence. So now we're getting at the other side of the question, which is, well, what happens to all of the FISH probes? Only two of the single-stranded molecules will base pair with the two molecules of chromosome, the two single strands of chromosome. So what happens to the rest? All of these other thousands, hundreds of thousands, millions of copies of DNA that are fluorescently labeled, ready to hybridize to a chromosome. What do? What do they base pair with? Themselves again. It's kind of a waste, but FISH probes are cheap. And they take care of the most important problem, which is that ensures that we use so many of them to ensure that they hybridize one of them, or two of them in this case. Hybridize to the part of the chromosome we're actually trying to detect. So that's FISH. Fluorescence in situ hybridization. PCR starts out the same way. You start with template DNA, that is the DNA you collected from a crime scene, DNA you extracted from a fish, you want to know what species it is, whatever. You heat it up, denature it. But instead of FISH probes, what are we adding to the reaction this time? What other DNA? This is primers, which are single-stranded. They don't start out as double-stranded like FISH probes do. They start out as single-stranded molecules. So they're already ready to base pair without being heated up even. There's lots of them as well, just like FISH probes. Compared to the number of molecules of template DNA, you have lots and lots and lots of these primers. The difference between FISH and PCR is that these primers, some of them are primers that point one direction on the chromosome. Half of them, in fact, will hybridize to one of the two strands of chromosomes. So let's say this is strand A and strand B. The other half of the primers you put in are a totally different DNA sequence. They would be complementary to the other strand of DNA. So what never happens, bottom line, for those of you that have been snoozing, what never happens in PCR is the primers don't base pair, we don't want them to anyway, base pair with each other. Because say this red primer that's pointing to the right, base pairs there. Because that's how we designed it, we control the DNA sequences of the primers. So we specify that this red primer that's pointing to the right be able to base pair with that part of the chromosome, and that primer base pair down there. So in this example, how long of a PCR product would we produce? How many nucleotides or base pairs long would it be? As drawn. How do we control the size of a PCR product? It's the location of the primers. So how big would this, how many nucleotides from one end of one primer to the other end of the other primer? This is 9 base pairs. It's way smaller than we would normally make a PCR product. So will those, to the question, I think, at the source of the question that was asked up here was, what happens to those two primers when you cool them down? Will they base pair with each other? Will you ever see something like that when you cool this mixture back down? Okay, so why not? No is the right answer. Usually the primers won't, and it's because they've been designed to totally different DNA sequences. So we could write in some example DNA sequences onto this chromosome. So if the sequence of the top strand is ATCGACTT, the primer sequence in red here is TAG, complement to the top DNA strand. What would the sequence of this primer be? Oh, I guess I better add another letter here. So what's true about the two black strands, the complementary strands of DNA? That's the easiest place to start. The sequence of the bottom strand, left to right. The top strand is TTA. What's on the bottom strand? AAT. Okay, so what's the red molecule's DNA sequence from left to right? TTA. It's the same as the top strand. Okay, so here's a great example. Will a primer that sequences TAG ever base pair with a primer that sequences TTA? Are they complementary? No, so they won't base pair. So this is a big difference between fluorescence in situ hybridization and PCR. In FISH, both of the strands of the probe will base pair with each other when you cool the mixture down. In PCR, that typically doesn't happen. Let's see, I saw a hand here first, then there, then.
So in PCR, you have a polymerase inside the mixture as well. So when it goes onto the strand, is it taking off the primers of the whole section or an individual?
So what happens to the PCR primer during PCR? It stays there. It becomes incorporated into the DNA strand. So here's some DNA. Here's a primer, base paired to part of the chromosome. So that's its 5' end, its 3' end, which means that what's the polarity of the black molecule from left to right? 3' on the left, 5' on the right, because they're antiparallel. So polymerase, the enzyme, comes in and starts adding new nucleotides from left to right and keeps extending this direction. So that primer is always the 5' end of the newly synthesized strand. It doesn't get degraded, it doesn't go away. It just becomes incorporated into the new molecule of DNA that gets synthesized in every step. Does that answer your question?
What about when it cools down and it tries coming back together?
Right, so then all of these are linked together. So every base pair is connected to each other. That's the job that DNA polymerase does. So when you denature this, the primer plus the DNA that's been synthesized melts, becomes single-stranded, and then in the next reaction, it serves as another molecule of template. So this is how PCR works. You start with one molecule of template, and then after synthesis, after one cycle of PCR, you have a second molecule that can serve as a template for amplification in the next round of PCR. So that's why there's an exponential doubling of the DNA in the tube. You go from one double-stranded molecule to 2 to 4 to 8 to 16 to 32 to 64, and after 30 cycles, which is about average for a PCR run, that's why you get up into the whatever 2 to the 30th power is, billions of molecules of DNA. So the primer becomes the template for future rounds of PCR. Question over here, sir.
Do you ever have to worry about the DNTPs binding to the primers?
So another thing that you add to a PCR reaction, besides template, primers, that would be 2, polymerase, so you have to have something for DNA polymerase to actually build new DNA out of, which is the nucleotides, A's, T's, G's, and C's. Typically called DNTPs for deoxy N, any, A, T, C, or G, triphosphates, TPs. So will these individual nucleotides, an individual half of a base pair, an individual base, hybridize to DNA? Little nucleotides floating around everywhere. Sure. They bind very quickly, they dissociate very quickly. So those of you that have taken or will soon take biochemistry will understand that in liquid, these molecules are binding, unbinding, binding, unbinding, depending on the temperature. That dictates how quickly NTPs, single bases, single nucleotides, will basically briefly attach to a strand of DNA and then leave again. So that happens all the time. It doesn't affect this process at all. So in the presence of NTPs, the building blocks of DNA doesn't interfere with the process of PCR. In fact, if you don't add those, you don't make any DNA because there are no nucleotides for polymerase to incorporate into double-stranded DNA molecules. Okay, there's another question over here. Yeah.
So with the probes, you could have single-stranded and double-stranded probes. And her question was, if the double-stranded probes would reconnect, why wouldn't you just always use the single-stranded probes?
So, to clarify, are probes single-stranded or double-stranded? When we create them to do FISH experiments, we create them as double-stranded molecules. They become single-stranded after you heat them. And that's the point in time when they can base pair with DNA chromosomes and show us which parts of the chromosomes contain the DNA sequences contained in the probes. And then when they cool back down, at the end of the FISH experiment, they become double-stranded again. So take home, bottom line, we put on way more molecules of FISH probes into a FISH experiment than actually hybridize to chromosomes. Because you only get one molecule, one strand of probe, hybridizing to one chromosome. Right? Human chromosome that's sitting there on the microscope slide. The other 999 million copies of this probe sequence, when you cool it down at the very end of the experiment, they base pair together, and that's it. So yes, the vast majority of the FISH probes that get applied to an experiment don't actually do anything. They're just there to ensure that the one FISH probe gets lucky. Successful, hybridizes, binds to the chromosome where it should. The same is true for primers. We put such huge numbers of primers into a PCR reaction to ensure that exactly the same reason. When we cool the template DNA down, we're sure that a primer is going to bind where it will bind its complementary sequence in the template DNA. All the rest of the primer gets thrown out at the end of the experiment, essentially. Other questions? Yeah.
So, do PCR and FISH produce the same thing? Do they serve the same purpose?
They're only similar in the experimental, what you do in the laboratory, heating and cooling DNA. So what's the purpose of FISH? Why do we use fluorescence in situ hybridization? We use it to find, let's see. Fish and chips? No. Maybe later. So what do these FISH experiments tell us? We have a probe that we know, this is a probe to a particular gene. We see funny colored dots. That tells us in which part of which chromosome does that gene exist in the genome. So FISH experiments help us learn something about genome structure, where genes are on which part of which chromosome, which arm of which chromosome, for example. So about learning about where particular DNA sequences are in our nuclei. What's the use of PCR? Sir?
Well, the FISH is to identify homologous DNA.
Indeed. So you identify homologous sequences. So let's see. Whoever visited me yesterday in my office hour already knows that I'm colorblind, so I'm going to give this a shot. Let's see. So I'm guessing that those are the same color, all of those dots. Is that true? Okay, good. So this is identifying the homologous chromosomes. You might notice that these pairs of FISH signals are at the same, they look like they're on the same end of the similar shaped, similar length chromosomes. So those are homologous chromosomes. If I drew an idiogram, this is kind of what those chromosomes look like. So those fish signals, whatever, what color is that? Is that green? Yeah. Okay, I'm using red, but that's kind of my drawn interpretation of those hybridization patterns. So those are the two copies, 4 molecules of DNA in whatever chromosome that is. Okay. Who else spots a pair of homologs? Yeah, these. So there's 2 copies of, well, 4 double-stranded molecules, 2 pairs of chromosome, whatever chromosome number that is. I don't even know if this is human, so hard to say. So FISH lets us find out where on a chromosome a particular DNA sequence, the FISH probe, is located. PCR is used for making lots of copies of a particular DNA sequence. That's usually for downstream applications like DNA sequencing. So you know you've got this chunk of a chromosome, you want to know what its sequence is. You make lots of copies of it using PCR first, and then you throw those copies of the DNA into a DNA sequencer, which tells you the sequence of the nucleotides in those fragments. So they both involve starting with chromosomes from a specimen, crime scene suspect, whatever, melting them, and then you hybridize something. In PCR, it's primers. In FISH, it's probes. Okay. This should work. Here's somebody's random PCR gel. This is to make a point that's a follow-up to the previous question. So what happens to all the primers? Throw in millions of copies of DNA into PCR. Primers tend to be about 20 base pairs long or so. They're short. They're single-stranded, so I'm not going to say base pair. I'm going to say nucleotides long. So how big is the fragment that is the PCR product in this case? I guess the authors have kind of done the work for us. If you can read the blurry molecular weight ladder on the left. And I might ask you this on Friday. What's the size of this piece of DNA? Yeah, it's presumably, because the authors labeled it this way, it's between 400 and 500 base pairs. So you compare the unknown to the standard, the molecular weight ladder, try to guess approximately what size in base pairs is that piece of DNA. So everything that's bright in this gel has been stained with a chemical because we've talked before about the fact that you can't see DNA visually with your eyes unless you stain it with something, some sort of chemical. We've talked about DAPI. We've talked about chemical banding to produce banding patterns. In this case, does anybody know has anybody done a PCR gel electrophoresis before? You know what chemical was used here? There are a few different ones. It's not one we've talked about yet, so it's usually a ethidium bromide. You don't have to know that. It's a carcinogen. So whenever you walk into anybody's lab, if they're using ethidium bromide, you should wear gloves. You don't want to get the carcinogens on your hands. So DNA gets, the bright stuff on here is all DNA. It's bound to a fluorescent molecule, probably in this case ethidium bromide. So that is the fragment that we're interested in at 450, 500 base pair PCR product. What's all of that? All of my students, the first time they do PCR is they focus on all that bright stuff at the bottom of the gel. That's where the primers are. Because there are 2 sources of DNA in the PCR reaction. There's the one template molecule you put in, or few, which you can't see because there's so few of them. You can never see that small number of DNA molecules. There are the PCR products. and then there are the primers. So that is what happens to all the primers you put into the reaction. The primers that are part of, there are primers that are part of the PCR products. There are some primers incorporated into those, but the vast majority of them, the smaller ones, small molecular weight because they're short, they run the fastest through the gel, they're at the bottom, they get wasted. You added them, they didn't get used to be turned into new molecules of DNA. They just sat there.
So you're saying the primers at the bottom are at the bottom because they're light in weight?
Yes.
But if it's supposed to go in this one, since it's going, isn't it going from left to right?
So where are the wells? Right, first step for reading gels. You can't see them in this case. Author has cut them off of the gel, but how in the world would you know which direction the gel's running?
Base it off the ladder.
So sure, the one thing to do is to base it off the ladder. So 500 base pairs is there, 400 is there. Smaller bands run farther faster, so that means they all started at the top, and the DNA is running that direction. So who knows, that's, I guess, 400, so that would be 300, 200, 100 is the bottom band in the ladder.
Yes.
Yep. So the primers are smaller than or about equal to or smaller than 100 base pairs, which is what we expect because, as I just suggested, primers are usually 20, 25 nucleotides long. It's the smallest piece of DNA you can visualize. It's there at the bottom of the gel.
Okay, then what is the 1 through 10?
Okay, so those numbers then, this author has numbered the wells. So those are the places where the DNA was loaded in the first place. They're saying there were 10 of them, and so they're running 10 different samples left to right. And only in wells 4, 8, 9, and 10, for whatever reason, did this PCR reaction actually work. In the other 6, for whatever reason, PCR did not actually successfully amplify DNA, which is why we don't see those bands there. So PCR didn't work. Yeah, maybe they didn't put template DNA in those reactions, so there was no DNA for the primers to amplify. It's one possibility. Talked a bit about FISH, a bit about PCR. We've talked about necessity and sufficiency. We've done some DNA structure, polarity, base pairing. Yeah.
Can you go for idiograms again?
Idiograms. Okay. So a bit of vocabulary and terminology. May I start actually with karyotype versus karyogram? Okay. So, did I spell it wrong? Apparently. IDI. So these are idiograms. What do they have in common? Are they pictures or are they drawings? They're all cartoons. They're some sort of artistic representation of chromosomes. That's what an idiogram is. So a scientist has taken a picture of a chromosome and then they've drawn a representation of it. In this case, in most of these cases, for example, in this case, the artist is trying to demonstrate a chemical banding pattern of human chromosomes. So that's an idiogram. So what was the picture that the idiogram was based on? What do we call that? That's the karyogram. So this is the picture I show you every time I ask how many molecules of DNA are in this picture. Right, so this is the actual original data, not a drawing that was taken, that was done after this picture was taken. These pictures were the originals. Idiograms are created from these. And then a karyotype, these are karyograms, gram, graphic, image. Let me pick one of these. What's the karyotype of that karyogram? I'm guessing that's human. So this part you don't need to know yet, but we haven't talked extensively about ploidy yet. Humans are diploid. We've got 2 copies of every chromosome. So the karyogram will start 2N. That's not going to be tested on this time. So during interphase, this individual has 46, well, should have 46 pairs in either 2X chromosomes, or they would be 46XY. What's different about this karyogram?
Only has a somatic.
So are all of the chromosomes, are there 4 copies, are there 4 molecules of every chromosome? Right, this is 1 double helix, 2 double helices, 3, 4 for chromosome 1. Same for chromosome 2, 3, 4, et cetera. Where do we stop that pattern? Right. Trisomy 21. So this is a karyogram of an individual probably that has Down syndrome. So their karyogram would indicate that they've got 1 extra copy of chromosome 21. But the karyotype is the written description of the karyogram. Does that suffice to answer that question?
So the karyotype is the fact that it's diploid or the trisomy 21?
The karyotype, depending on what karyogram you're looking at, the karyotype will change. So in a normal human, female would be 46XX, male 46XY. I don't remember the details of how human cytogeneticists write the karyotype for Down syndrome. Something like 47 tri-21, trisomy 21. They have a shorthand notation, which, since I'm not a clinical cytogeneticist, don't remember offhand. But we could probably find out. If you needed to on an exam, you could too. Let's see, what happens if we Google karyotype Down syndrome? And don't do images. Wikipedia, that sounds useful. Does anybody see? There you go. So that's the karyotype for Down syndrome. So 47, so 46, 2 pairs of 23 plus 1, 47, 2 X chromosomes, and then plus 21. So the plus 21 indicates that one of those 47 chromosomes is an extra plus copy of chromosome 21. Don't need to know that for the exam. Now that we've covered it in class, it's going to be on the test. This is to remind you that you can do this during the exam. If you need to look something up, go for it. Please, as the instructions I forwarded on Blackboard suggest, if you do use an external source that's not your notes from class, just please tell me what source you used. That way I don't call it cheating, I call it you were using your resources to your best ability. So when you use an external data source, just tell me at the bottom of the answer. I looked some of this up at the Wikipedia article for trisomy or whatever. That's all I ask. Yeah.
In karyogram, are the chromosomes
chromosome always in the double form or in the single form? Is it always 2N or can it
So
be one?
the number of copies of the chromosome in a karyogram depends on the species we're looking at. So for example, humans are diploid. We'll always see at metaphase, there will always be 4 copies of four double helices for every chromosome. Right, again, just to recall, that's 1, 2, 3, 4 double helices, and that's all just chromosome 1. So metaphase, every individual has 4 copies of every chromosome. This is for a diploid. There are some species that are tetraploid. They've got 4 copies of every chromosome. That's like salmon. A lot of plants are tetraploid, some are octoploid, some are dodecaploid. Some strawberries have 12 copies of every chromosome. I don't know, plants are cool.
So on the
So it depends on the species you're looking at, how many objects, how many molecules of DNA you'll see in a karyogram.
So on the karyogram of human, we will always see a diploid chromosome.
Right, so in humans you should always, unless there's a mutation, some sort of chromosomal rearrangement maybe, or an extra copy of a chromosome like in Down syndrome, then you'll see an extra something or not enough something. So that's why, for example, in this case, Down syndrome, you see extra chromosome 21s that aren't there in most humans. Yeah.
So these would be examples of homologous chromosomes, correct? Because each one is condensed so close together that you can't see the sister chromatids? Or
Okay, so yes, good question about terminology. Sister chromatids and homologous chromosomes. What stage of the cell cycle is this? Are all of these taken at?
Metaphase.
Metaphase. Okay, so at metaphase, what we're looking at here, let me pick on chromosome 4, which now you really can't see, but that's okay. I just picked it because there's a lot of white space next to it. So that object right there, it's 2 double helices next to each other. I know I've done this before, it's just refresher, this is the review session. And so the one next to it, also a double helix. So that's what we're looking at right there. So metaphase, diploid organism has 4 copies of every chromosome. So that's what we're looking at in a karyogram. So which are sister chromatids? So are these 2 things I'm pointing in with red arrows, are those sister chromatids or are they homologous chromosomes? Trick question. Go ahead. So we've got to vote for sister chromatids. Uh-oh, now I told you it's a trick question. It's both. Every double-stranded molecule, every double-stranded DNA is a chromosome. It's a chromatid. So you could call these 2 chromosomes. That would be fine with me. But at metaphase is the only time we call them chromatids. There's a historical reason, and it's unfortunate I've already apologized for it on behalf of the geneticists that came before me. So at metaphase, we call chromosomes, double-stranded molecules of DNA, chromatids. So here, circled in red, are 4 chromosomes. You could also say there are 4 chromatids. Either is fine. They're all homologs. So we'll end with this. Got a couple more minutes left. What's true about the 2 chromosomes that are sister? That would be the two that are actually physically connected together there at the centromere at metaphase. Sister chromatids.
Identical.
They're identical. So sister chromatids, the two that are held together, they're identical, which means that they came from the same parent. So what this is, and we'll look at this a little bit later, next chapter perhaps even, those two molecules of DNA are, for example, in this case, chromosome 4 that came from your dad. This is my nomenclature, not genetic nomenclature, and a copy of it. There's chromosome 4 you got from your dad at fertilization, there's two copies of it. What are the other two sister chromatids of chromosome 4? Right. So you've got chromosome 4 you got from your mom. Again, the M's and D's, this is my nomenclature. And its copy, chromosome 4 from mom. That's why they're called sister chromatids. One of those 4 M's was just recently synthesized by copying the other version, the other chromosome 4M. And the same is true for the two 4Ds. One of them was the template, the other one is the new copy. So put simply, every time you look at a karyogram, you're looking at two copies of a chromosome from mom, say, and two that came from your dad. So the sisters are genetically identical. The homologs, one from mom and one from dad, are pretty close to similar, but are your mom and your dad genetically the same? No, they're different people. They've got different mutations. So the chromosome 1 pair that you have, there are differences between your chromosome 1 you got from your mom and the one you got from your dad. Unless there's inbreeding going on, but we don't have time to go there. So it's office hours now, and I'll stick around and answer questions, then we're going to head over to my office.